All right. So this is a series of surprising things. Thank you. So a bunch of surprising findings that we made, and it'll be cool to get your feedback about this. So what I'm now going to talk about is the work done by this wonderful set of people who are in the lab now as well as before. And let me skip all this and come right back to this. So there is this brain region called hippocampus. It's kind of in the middle of your brain. It's about that big, about 2 thumbs, so pretty tiny. And people found that if you have damage to hippocampus, all the way in the 1950s, and Brenda Millard made this discovery, that hippocampus is required for episodic learning and memory. Such as right now, I'm seeing a few words, you are seeing some lights, and we'll see how different parts of your brain respond to light and so on, but the idea is that eventually it all ends up in hippocampus. So that's roughly the wiring diagram, quite old, but still quite relevant. So right here is the retinal ganglion cell, just the last part of the station in your eyes. From there, it goes to the one little nucleus or area called thalamus or LGN. From there, it goes to primary visual cortex, where you have this Gabor patch selectivity, and then a plethora of brain region from which the signal keeps traveling. And then all the way at the end is this region called HC or hippocampus. And if that hippocampus gets damaged, you have problems remembering conversations. You don't remembering episodes. You don't have problems seeing things. You see things quite well. But you have problems remembering sort of episodic memory. And we got interested in that to say, how does it work? So let's start looking at it. So in the very first station in the brain, as the research of Hubel and Wiesel showed, neurons respond to these oriented bars or Gabor patches. Very distinct concrete stimulus that gives responses. But on the other hand, when you have hippocampal damage, you can still see these oriented bars, which is quite an interesting abstraction out of some random stimuli. And then people have all these diseases are involved in hippocampus damage. So Alzheimer's disease literally begins in part of the hippocampus. Autism spectrum disorders, kind of intellectual disability. Intractable epilepsy leads to serious learning deficits. Depression interferes with learning. So it's a funny thing, a tiny region like that is somehow responsible for so many diseases. You take a random part of neocortex, you don't have so many diseases in any part. And this happens with the oldest part of the brain. So what's going on? Snakes have this part of the brain. Snakes don't have all these fancy neocortex. So that's a circuit. And as I mentioned, the discovery of Hubel and Wiesel in 1960 showed that neurons in this part of the brain, primary visual cortex, the first part of the neocortex under your skull, they respond to this. That's my phone, believe it or not. That serves me right.
It's like
Oh no, it's not me. It sounded like my phone, but it's somebody else. All right. Maybe it's still me. So then people got curious. So, all right, how the hell does this come about? Where does this all fancy stuff come from? Like depression and memory and episode. Right here is pretty concrete stuff. So it got people's attention for the longest time. There's something to do with intelligence in this whole circuit somewhere. Where is that? So that's where, as you know, these fine gentlemen, John Hopfield, started his work there. And he said, let's study hippocampus, because there are neurons in hippocampus. They have recurrent connections in a region called CA3. So he came up with this RNN theory based on Ising model that got the Nobel Prize. And then Jeff Hinton looked at this entire circuit and said, aha, I can do error backpropagation, and this circuit will give me interesting stuff. So lots of cool work. And one important fact to remember is that the Nobel Prize is actually in physics, not in medicine. Computer science didn't exist when TNT was discovered, so that doesn't happen. So the race is on. There's lots of cool stuff happening. But how do we understand how the damn whole thing works? How do we understand that? How do we go from this to episodes? So people did this work long ago, and they found that if you go in retina, the neurons in retina don't just act like a little pixel. They do bright light, dark light. These retinal ganglion cells respond to either bright centers surrounded by dark or dark centers surrounded by bright. Neurons in, so that's this retinal cell, the first station in the network. Here's the second station in the network. They respond to like something more complicated, which is just a series of these dark and bright patches lined up. And there's some evidence that's happening. And then once you line up this bunch of bright circles divided by dark, you get something like a bar. And when you line them up, you get something like this. So that's reasonably well understood. But then people went on and said, okay, let me flash these Gabor patches. Look what happens in hippocampus. Zero. Nothing. Nada. No response. So what the hell? So people said, all right, something is going on. And people started to futz around. Say, okay, where is the episode? You turn on the bright patch, turn on the oriented bar. There should be some episodic response. Nothing. So then people were futzing around, and they made this amazing discovery that hippocampus is actually the GPS system of the brain. Like, wait a minute, I had oriented bars, something I understand. All of a sudden, I ended up in GPS, self-driving machines that we are. So let's listen to what they do. So this is Nobel's speech for the pioneering discovery of John O'Keefe. So he's going to demonstrate a typical experiment that we do. So there's a rat that's going to be running around on this platform. It's about that big. And people are throwing little bits of chocolate. And he just like a chocolate vacuum cleaner just runs around, picks up bits of chocolate.
If we place an animal into a circular environment, we watch
so it's just running around.
Using an overhead camera.
And each dot is a spike. As everybody knows, neurons generate individual punctate spikes. So all these spikes are lining up here. One single neuron out of about 500,000 in the rat brain. They don't spire anywhere else. And from there, you can generate this kind of a heat map.
And so you can see all of these red dots are adding up, and which I'll say very little about.
Oops.
Which I'll say very little about because I'll leave it to the.
I don't know why it's doing this.
Which I'll say very little about because I'll leave it to the Moses.
All right, we'll have to watch this again. Sorry about that. I wanted to speed this up. It's taking too long. I want to skip this. All right, so let's be patient a little bit. So we're going to see that experiment again. The rat's going to run around here. And from there, we're going to come to this heat map.
By my colleague Julia Krupich. If we place
an
animal into a circular
So
environment,
quite funny, it's not just that every neuron fires here. Some neuron will fire here, some fires there, some fires there, and so on. The different neurons fire at different places in the maze.
And it's only when the animal goes
by looking at just 100 neurons, you can tell where the rat is with that much accuracy. So, well established.
And so you can see all of these red dots are adding up. And what we typically use is
So then we turn that into this kind of heat map.
Represent the firing rate of the cells. So the heat map shows that this cell fires with a maximal firing rate of 7 hertz here, and successively lower rates as you move away from the center. And this is the typical type of way in which we represent these cells. If we look, and one thing that I should point out is that if an animal runs around in an environment like this in an open field, it turns out that it doesn't make any difference which direction the animal runs through the place field. If it runs north, the cell is happy to fire. If it runs south, it's equally happy to fire. It looks as though the cell is representing the location and not something about the sensory impressions that the animal is getting in that location.
So there you have it. We started with neurons responding to very specific aspects of stimulus. And then you go all the way at the end, these neurons no longer respond to any specific stimulus. They're just responding to some abstract location. Now, how did I go to location of all the things? Because I can perceive the world and I can perceive episodes without going anywhere. In fact, Alzheimer's patients, they have this serious disability remembering a conversation while they're simply sitting there and you're sitting next to them. So where is space coming into all this? So this led to lots of heated effort by lots of people. And then the effort led to a big surprise. Lots of theories were built, starting with Hopfield and many others, about how that abstract spatial representation can come about. Then people thought, okay, the same thing should work in our brains too, because we are the ultimate abstractors, perhaps. So they put electrodes in monkey brains, pretty close to us, since human beings don't want to have electrodes in the brain and run around unless they have serious disabilities. So put electrodes in the monkey brain and let the monkey walk around. Same experiment like this. And zero spatial selectivity. So neurons do not care at all for where the monkey is. So that was a big oops. That study came out in 1996 by Edmund Rolls. And that was one study. Yes, please. Oh, you put tons of electrodes. So now people, there's no one neuron. This neuron that I'm showing you right now, you just stick an electrode and 80% of the neurons will do this. So you don't have to select anything. You just stick an electrode. 80% in biology is a lot, right? Kind of like 70, 60 will be quite a bit too. Primary visual cortex, the number of neurons that respond to Gabor patch is like 40 to 50%. So 80% is quite a lot. People put hundreds of electrodes. So Edmund Rolls found this, no place cells in primates. That was a bit worrisome. But he said, on the other hand, neurons depend on what the monkey is looking at. Whereas in rats, they don't seem to depend at all on what the rat is looking at. So that's the opposite. It's not like the electrodes were in the wrong place, or neurons were busted, or monkey was confused. There was selectivity, but not to what they saw in the rats. And then people did more experiments. They put electrodes throughout the hippocampus, hundreds of electrodes, nothing. 5 to 10% at most, kind of like next to chance level. So the race was on. What the hell is going on? And then a bunch of people from UCLA got involved, and they said, maybe hippocampus has something to do with memory, so we should show the images of things that everybody will know. They showed images of like famous people, like Jennifer Aniston. Back in the days, that was the famous people. Maybe even now, I don't know. No offense to anyone. And then they found that individual neurons responded to the just face of Jennifer Aniston comes in, and the neuron responds. So now what the hell is going on? Is it like a different algorithm in case of the brain of a rat versus a human being? And how did we go from just a Gabor patch, which none of us have really seen? Yes, please.
How big are these electrodes compared to neurons?
They are just the same size as the neural cell body. Good question. I can go ad nauseum into the electrodes and so on, but for now, just take my word for it that to the best of everybody's ability, all kinds of techniques, you get these results.
So it's, I can
10 micrometers.
We can think of it as a single neuron.
Yes, yes. For all practical, this is like, if that didn't happen, then we have to throw away all of systems neuroscience. So it can happen, but I think right now it's good. Lots of complicated electrodes. We don't use just one electrode. We use a bundle of 4 electrodes. And we do triangulation, so we separate many things. We look at waveforms, we cross-validate. So it's a solid field, and I would say it's pretty reliable. It's not, the problem is not, you're absolutely right, that the neuron, you are mucking up 3 or 4 neurons, and now I think it's garbage. If that was the case, you will not say selectivity to what the animal is looking at, so it's not exactly that. Nor will you get Jennifer Aniston cells, because now, you know, and that cell doesn't respond to all famous people, responds only to Jennifer Aniston. Other neuron responded to Eiffel Tower, and third responded to some other actor or something. So it's kind of not just mush out there. So now we have a problem that we have been using this system for all kinds of things, including clinical research, where we make animals with Alzheimer's disease, give them a drug, see if the animal is better or not with the drug, and what's the test? You can't ask the animal, hey, what did you have for lunch? How do you ask them? So you say, okay, go run around and solve a maze. That's a test we use, because we think that with place cells, you're going to find out where you are. But in monkeys, there are no place cells. Instead of that, there are responses to specific objects, and in the damn deep network, you started with oriented bars and dots of light. So what the hell happened here? So big puzzle. So I'm going to jump through a whole bunch of things because this is going to be a rather short talk. So pardon me as I zoom out of this thing and so on and tell you directly the results. So what is it that we did? And I'm going to skip a whole bunch of things. As you can see, we have been busy. So just to impress you that I'm not just talking random stuff. So we said, all right, let's first see what the experiment is. So let's kind of magnify everything, because who knows what rat perceives. So we made, okay, all right, the rat's running around in this platform, even though, because when the rat runs around, his eyes are right next to the ground. Right here is his eyes. So he can see tiny things very, very amplified. So maybe he perceives, even though we keep it very smooth, so this is the ground. And then the table is above the ground, so the carpet, we exemplified, so has some weird features. And on the walls, we put gigantic stimuli, fractal-based something, so that it's something he's never seen. So we don't have to worry about he hated it, liked it, and so on, just generate by algorithm. And now if you say, where is the rat in the maze? Well, you'll say, all right, if the rat is here, then I can tell you where. If you ask me, Mayank, where are you in this room right now? I'll say, well, I'm like 6 meters to the left of that white wall and 10 meters in front of the brick, and that uniquely defines my XY coordinate. So that's what will give me. If that's the case, the neuron should depend on what the hell the animal is looking at, because when I'm looking here, I describe myself like this, and if I look here, I describe myself as completely different. So how come rats don't have this? So then we thought, wait a minute. There'll also be smells. And rats have a phenomenal sense of smell. Their nose is next to the ground. People in Nigeria use rats to detect landmines 0.5 meter underground. So that's an amazing sense of smell. We can never imagine what they have. There's big fat brains. That's clearly everywhere. We can't control it in real experiments. And then there are sounds. Right here, ultrasound. If you pay attention right now, there is a sound source I can tell, which is somewhere here and not there. And if it were to go off, you'll say, oh, something happened. So who knows? They may pay more attention to that. When they're running around, and primates may not, they have internal organ. They move the head, the vestibular organ. And the biggest stimulus of them all for the rats is the tiny bit of chocolate that's falling on the ground. That's the only thing he cares for. Otherwise, why the hell will he run around with a bunch of electrodes sticking out in his head? Right? So that's why he's running around bits of chocolate. Right? We will go quite far for chocolates, and so will rats. So what if these neurons are responding to some stuff on the ground, which primates and us do not? Because our heads are 0.5 meter, quite far, and we don't have big olfactory bulb. So how do you test these things? So we said, all right, we build a virtual reality for a rat. So now we make rats behave like us, but this virtual reality is made for rats. So we don't put goggles, because they don't realize, oh my God, if I don't see my hands and feet, I'm going to freak out. So we don't want to do that. We don't want them to stress out, because the goggles can stress them. We don't want to head fix them, because that's stressful. So what we did is that we put a little vest on them. Like babies like to get swaddled and calm down, we put a similar tight vest. So I routinely say our rats wear tuxedos and nothing else. That's the pretty good life, and eat chocolate. So good life. And then we put a little Velcro so they can stand up. As they run around this flat ball, there's a hovercraft mechanism that goes from here to a VR engine. That engine goes to a pico projector. Our astronomy friends did a polished mirror, which is pre-distorted. So the image falls all around, including up to his feet. So now he's walking in a world where there are smells here on the ball, there is texture on the ball, but it has no relevance to where he's going. He has to rely only on vision to go somewhere and get a reward. And if he goes to the right place, then he gets a little sugared water, and just like we like soda and so on, he likes sweet water too. So we can now do crucial thing. We are making the rats behave a bit like human being. The non-specific cues are uncorrelated behavior. There is smell, there is texture, the brain is intact, but they're just made irrelevant. So it's to pay attention to vision only. We can do many trials, we can reliable electrophysiology, and now we can test rodents, primates, and humans in the same condition. Because this is built in such a way that you can take the whole thing out, put it around a human head, such as my head. I have done this experiment, and I'll show you several tasks that the rat do. It's quite difficult to do this virtual navigation task. So let's see what the rat does. So he has to now go underneath this pillar to get a bit of reward. Somehow the audio is gone. Yeah. And when he reaches there, we give him a beep to say, yep, you've reached there, like a kind of like a chocolate sound, but not a real sound. Otherwise, he gets confused. Then the pillar goes here, and you can watch him. He's associated that pillar with the reward. So now we have associated some stimulus in the world with reward, which is important for reinforcement learning and so on. But it's a clear stimulus. And now the pillar is behind him, and he reaches the edge of the table just based on motion parallax. He figures out he's reached the edge. Even though his whiskers and his nose doesn't tell him anything, he does this. So pretty cool. He does this many, many times. And now, what happens to these neurons? Because he's doing the task purely based on vision, so let me jump through and show you just one neuron in this, and then we get going. I don't want to show too many things, otherwise we'll be here forever. So let's just listen to one neuron's activity, all right? So now, in this world, the rat's running around just like the real world, and we're going to render his path. So he's running around, this neuron is active. Fires a huge burst of activity here, like John showed. But then there's a different burst somewhere else. But notice it's a long burst, 2 seconds long. So just kind of garbage, right? There's nothing here, except there is delta t. Whenever the neuron is active, it's active for about 2 to 3 seconds. I don't know when you speed it up, you could see it, but we did the analysis in this. And what we found is that actually, this neuron in this case is counting delta x. How much have I moved with respect to some signal, which I will tell you later on. It has abstracted out how much have I walked. And then if you take the rat and put him in the real world, that neuron will fire for the same 2 to 4 seconds in the real world too, except all those 2 to 4 second long bursts are in one spot. So we forgot the fact that when we are behaving in the world, our perception is rather slow. And we know that our world, under majority of the conditions, obeys continuity equation. The world changes somewhat continuously on the scale of 1 second, and this region is picking that up. Compared to the speed at which I'm moving, everything in the world is changing very slowly. And if you look more quite carefully, you will find the same thing for the same neuron. That if the rat were to run in the real world, this is the path of the rat in the real world. This neuron has this somewhat long place field. If we run in virtual reality, these bursts are all over the place. But whenever these bursts occur, they're about that long, about 2 to 4 seconds long, just like this. So since I'm a physicist, we call it the string theory of rats. There are little strings here, right? It's real. String theory is real. All right. So now we got one ingredient to say when we are behaving in the world and when we want to make something like episodes, maybe the first thing we are doing is not responding to just a Gabor patch, but a little short segment of time. Is that really true? Can we actually check that? So we did one experiment. So let me see if this network can actually do abstraction where the fundamental element is not space or anything else or not Gabor patches, but simply the passage of time based on what I did. So we did that experiment, and that comes out pretty well. So this is simply to show that this matches. So if you looked at this neuron in virtual reality, no spatial selectivity. I didn't show you, but neurons care for which direction. And you find the same thing in primates. In the runaround, spatial selectivity is only 5%, and the direction selectivity based on stimulus is 15, 16%, rest is nothing. So it kind of matches in primate to say vision alone. We conclude from this, I didn't show you this part because we are out of time, so let's focus on this, that vision and locomotion are somehow combined in the brain, how we are going to discuss, to generate distance selectivity, delta x, but not where you are. That something else is required. So can I now get into abstraction out of this? Because now I have removed all those smells which are maybe causing place cell. If there's one smell here, the neuron may fire here, other cell here. So there's like a whopping stimulus that's taking out some interesting stuff that this network can do. So we did one more experiment, and that's where magic started to appear. So watch this carefully. Probably the most interesting thing I'm going to tell you is in this little video. So he's going to run around in virtual reality. And you get this long burst, like I tell you before, about 2 seconds long. And then another burst as well, seemingly random. And yet another burst. Instantly, it did abstraction. Instantly. What is that abstraction? Say, notice that this is an equilateral triangle. At every location, the visual cues are very, very different. And what I'm going to show you is that individual visual cues do drive the hippocampal neurons. I'm going to show you which ones. But in this case, there was consistency. So whenever he started, he had just obtained the reward. That's why he stopped here. He just started to accelerate, his internally generated sense of acceleration, and the visual world was coming at it from a long distance. So all three things were consistent, and the same consistency happened at all the three places. And now neuron, instead of generating a place cell, actually generated a distance selectivity very, very precisely. It got locked in. So the algorithm in the brain seems to be, I haven't proven to you yet that vision plays a role that I'm going to show you next, to say if my behavior in the world is something random, such as I'm walking around randomly, then there's nothing consistent except that I'm walking. In which case the neurons generate delta t/delta x. Both they do. Each neuron prefers certain delta t, and that delta t or delta x, some neurons prefer delta t, other delta x. As soon as there are things consistent, it starts to encode that. And if nothing is consistent, it just says delta x, and I don't care where I am. Yes, please.
Is this thing dorsal CA1?
Dorsal CA1. They're all regular place cells. We take the rat out here, put it in the real world, they look like nice place cells. But that's because there are too many things driving and we are not controlling them. All three the same thing. So now we got something. Then we made all kinds of paths. This neuron is another one. He's going back and forth. Now instead of triangle, this is a triangle path. It's not exactly triangle, but still it's abstracting out. This neuron fires at both ends. This neuron does another AND gate rather. It fires in this direction alone, but not that direction. So its abstraction is not just distance, it's distance plus direction. So this says distance plus direction. This is pure distance. This thing is distance and direction. This is distance and direction on two arms, but not the third arm. So it's extracting lots of interesting features. Just because you remove those giant proximal cues, now the calculations are doing interesting stuff. So, yeah, please.
Would more ventral cells have longer time horizons?
Yes, absolutely. The cool part is that in the dorsal CA1, some neurons will be half a second, and other neurons, the same tetrode, 5 seconds. So they are doing a variable nature of delta t and delta x rather than just a fixed one, which can get fooled. So now we have shown that depending on the nature of the experience, the neurons are abstracting out different things. If they are running around purely randomly, it's just delta t or delta x in virtual reality. If they are running around on more or less systematic paths and not perfectly, it instantly abstracts out distance and angle in variable ways. It doesn't fire notice in all these places. It cares only for this. And if there are proximal cues present, just forget about all these complicated calculations. I'm just going to respond to the stimulus and gets locked in here. So now we figured out, and there are lots of studies to say the same neuron can either encode delta t or delta x, we're just going to call it time, or space based on multisensory olfactory cues, or distance and angle. And then we got excited to say this must happen in primates too. So we made a prediction that when primates run around in the real world, they don't have place cell. And that's because they're more like virtual reality, only vision. You find something like this. So we predicted if you now take a monkey, and instead of walking around randomly, you make the monkey walk around a linear path, locally linear, you should get back this selectivity. And we looked at old literature, like 99 and so on, and that's what they found. If in an experiment the primate was walking around on a linear path on the perimeter rather than all over, you instantly got the consistency detection. You instantly found what looks like place cells. It's not really place, it's actually consistency detector. So we got some hook into this to say, what the hell is the link between these things and so on. So I want to share one or two things before, but are there any questions before I keep rushing through? There's a lot of complicated material. Yes, please.
So when you say linear path, what do you mean by the linear path?
Meaning it's walking around on the same path again and again. Notice in the middle, the monkey doesn't go that often. It has the same experience reliably again and again. So when
it doesn't mean that it means in just one direction. It's saying that it's somehow unique.
At a given location, the number of distinct experiences are rather limited. So when I'm at this location, in a random foraging, sometimes I'm accelerating this way, sometimes I'm decelerating that way. So those, my self-motion cues and visual cues are not consistent. But if I'm simply going back and forth here, then I'm always either going this way or that way. Either I'm accelerating or decelerating, but there are only two values rather than huge number of values. So in that case, because the monkey is walking around in this linear path like this, he walks only in this, and he can either go this way or that way. Instantly, the selectivity appears, so this system is detecting consistency across multiple things. Did it make sense?
But
Not
is
really?
it something that happens because of the experimental design or doing nature?
That's right. Now, so this is a great question. So we start to look ethologically to say when animals are finding their ways, elephants are finding water from 100 kilometers, where at least people claim 100 kilometers, but I definitely believe 10 kilometers. They can smell water from 10 kilometers, so that gives them sense of direction. And then they follow systematic paths. They're not like a straight line paths, but they follow systematic paths through this thing by using various visual landmarks and so on. So there's evidence that animals are not just wandering around randomly, and something like that is happening in nature as well. But I'm going to come a lot more to that in a minute. Yes, please.
So rats don't have as good a visual system as primates, but did you think that the consistency for the rat was also based on visual cues? Because they're really
Good question. I think I gave you some money to ask that question, right? Because that's the next. Oops, I revealed it. So that's the experiment. That's exactly what we went after. So what if rats are different than us? Their vision is crummy. They don't care. It's all self-motion. This much matches, but in biology, lots of things match. Who cares? So we decided to do an experiment. Lots of experiment, but I'm going to skip all that and how it works and blah, blah, and so on. Let's go to skip all this and go to this experiment. So we got two very bright people, and we said, let's do pure vision test in a rat. So very nice, brilliant people, Chinmay and Shunali, and the experiment is even simpler. So we removed the olfaction and whisker stimuli and so on by building virtual reality. Now you say get rid of locomotion and get rid of rewards too. Because when you're listening to me right now, hopefully there is no punishment and there's definitely no any reward. You still remember something, right? So no rewards, right? So we do an experiment that could have been done by Hubel and Wiese, right? So a rat sits there, you make a very salient bar of light, green and white, green and black, it just goes around, that's it. Now, all that happens is there is statistical regularity in the motion of the bar. And that will activate retina and everything else. Let's go all the way till the end. So he said, all right, the rat sits down here. This is his field of view. Our view is roughly this much. Rat's is roughly that much. So this area he can never see, almost never. He can cheat, but he doesn't care because there are no rewards, there is no task. He just sits there. There's a big stimulus. This is drawn to scale. And this green and white bar just goes around. And just to make sure that the experimental conditions are the same, we gave him random rewards so that we didn't like one experiment was with reward, other was not. So we just gave him random reward. So he remains sort of alert or God knows whatever. And now, if his vision was poor, there should be nothing. That's one neuron, which if he walks around in the world is a nice place cell. These are different. This bar of light is when the bar of light is going counterclockwise. Only this neuron fires whenever the bar is at this angle with respect to his head, at around 90 degrees, and these are 60 trials. Interestingly, the neuron doesn't fire for many trials, long story, and then very reliably fires at that location, nowhere else. Now, you cannot say it's anything else. It is vision, nothing else. Here's another neuron that fires to his left slightly, around this direction. There's a third neuron that fires here at this direction, more or less. So these neurons care for what was the angular position of the bar and the direction of movement. They're abstracting out two statistical regularities. One is the position of the bar, where it is going from the past, and other is which direction. Yes, please. Oh, colors are simply to show that it's from here to there. So central colors are blue because it should be wrapped around, right? So we want to make sure that we don't say this is different than this. It should be a cylindrical plot, but we can't make cylindrical plots. So we're putting identical color here and there, which we can map here. So none of these neurons are active when the stimulus is here. That corresponds to the pink, and the front corresponds to blue, and so on. Okay? And then there's another neuron that fires all the way when it's almost at the periphery of his vision, very reliably. So similarly, seemingly just a stimulus response, but then the final neuron, this fires nothing when almost nothing when the bar is in front of him, very reliably in the region where he cannot see at all. So the system is able to calculate. If you did this experiment in visual cortex, which somebody should do, you might not see any of this. So now it's doing visually driven as well as some kind of abstraction. And notice the duration of activity. Once again in this, almost a second long. This thing takes about 5 seconds to go around. So these responses are once again have the typical time scale of a second or so. So keep that in mind. So this keeps going on in this area. That sounds pretty interesting. And we did a lot of other things. So at this point, we want to go back to the full abstraction. Can we do that? So instead of now going down the path of simplicity, now let's go to full task. So we did two different things. Yes, please.
Is 0 when they have no experience, and so are they learning between 10 to 30
No,
tracks?
they've done it every day.
Okay.
We just did it every day because we want to space. The rats have tons of experience. So we want to compare with this. So they've been watching this or not watching this. We don't care what they do for like a month.
I see.
So they've been seeing it.
So this is overtrained rats.
Oh, I won't say trained. It's just that we presented it. They don't do anything with it. Maybe they fall asleep. Sometimes they do fall asleep. Sometimes they run. They do weird things, but there is no training. It just
Overexposed.
Overexposed. Yes, thank you. That's right. These are overexposed, right? Just like space that we live in, we are overexposed to space because right from childhood we are walking around. Those are interesting questions. What will happen in the other case and so on. So then let's go back to what happens when the rats have to actually navigate. So we say, all right, now let's bring up the whole thing. And that's an interesting title. Do the legs know what the tongue is doing? You have to read the paper to know what the answer is. So now rats back in virtual reality. Sorry, let me pause this. Yes, please.
Just a quick question. That you showed one of the neuron fires when the rat doesn't actually see anything.
Yeah.
Right? So it anticipates.
That's right.
It knows the cycle.
That's right.
Anticipates.
That's right. We think so. And so we got quite excited by this. So this was just angular direction. So then we took a linear direction. So bar of light simply approaches, which means gets bigger, and then goes behind him. He can see it go behind him and goes forward. Now the neurons once again encode which direction the bar of light is going and whether it's approaching or receding and exactly at what distance. Once again, roughly 1 or 2 seconds. So the delta t or delta x is preserved across 5 different experiments. Pretty fun trick. So that's kind of basically consistency detector is the main thing by which we seem to learn. Kind of like what we do in language models too, where we say what is the most likely answer. This is the first one. But now what you will see that when you make the system do something very difficult, then it shines in full color. So let's do that. So now he's not just sitting around chasing little bits of sugar. He has to actually map this space. So we put him in virtual reality. It's a big maze now, 2-meter diameter. And he has to go to an unmarked location, and if he goes there, he gets a reward. There is nothing that tells him what that is. He has to combine all those visual cues and locomotion cues to figure out where he is. So let's listen into that. And we put sounds as well to make it more natural. So he's facing the wall now. He's going to turn around. It's kind of difficult, so he has to learn that he has to turn around, and he figures out to turn. And it's a hard task. I invite you to come to my lab, or our lab. You will fail. I have failed many times. He reached exactly the spot. He's overtrained. He's off by just about 5 centimeters. So remember that spot here. He's off by just this much. He gets stressed out. So he grooms himself. Like, what the hell? I don't know if he thinks that, but that's what I would think. Like, what the hell, dude? Give me my chocolate, sugar. So he figures out something, runs, and notice he'll going to hit that same spot. And he's going to hit there. And then we give him a feedback to say, yes, you got it, get sugar water, teleport him to another spot. So no human beings, no clever hands technique, and so on. And he does this very reliably. Second direction he goes. So we basically release him from, this is classic so-called water maze, but using here positive reward. He either is released from here facing the wall, or here, or here, or here in pseudo-random fashion. And you can see when he goes from here, he kind of makes a beeline to this spot. When he goes here, he kind of goes there. The paths are very distinct. So he knows what he's doing. So how does he do that now? Because now he has to get a reward at a certain location based on some features of the environment and his locomotion. So we looked at it, and the classic idea was that we'll get place cells, and that's what I was looking for. Nothing. Here's one neuron. It's kind of all over the place, just like random foraging. So the fact that the rat had to do the task, make actually map of memory rather than just choose a pillar, the cognitive demand did not bring back what we were looking for. Didn't work. And this is true for most neurons. So at that point, Jason, now who is in job market, so hire him, we thought, wait a minute. If I had perfect place cell, it's like in quantum mechanics, if you know where you are, you have no idea where you're going. It's not enough. If I had neuron that tells me exactly where I am, and it's an abstract location, how the hell that will tell me where the reward is from here and which direction I should go, how far should I go, and where is the location? So we thought, okay, maybe that's what we should code up. So instead of using a complex AI model, we use the generalized linear model. We cross-validate this. So in the generalized linear model, say, all right, tell me if the neuron either codes for distance travel from all the start point, distance is distance, based on when he started to run, not on anything else. So-called episodic distance, or the angle with respect to hidden zone, and the location of the reward. These are the three things. If I know, I can do the task. So we look for it, and that's exactly what we found. So here's one neuron. The activity is color-coded based on the distance travel. This is just distance travel on x-axis, regardless of start point. Notice if they were place cells, they will fire either here, or here, or here, or here. Instead, they fire the exact same distance across all the whatever several dozens of trials. This neuron fires reliably at 10 centimeters from the moment it starts to run. There's another neuron that fires reliably at exactly 50 centimeters from the moment you start to run. This is the distribution of the preferred distance across about 200 neurons. Most of the neurons encode about first 50 centimeters of his distance because that's the most likely distance he has to travel in a 2-meter maze. So that's the first thing across the ensemble it figures out. Second, the neurons encode the direction in which the reward is. So now here the spikes are encoded with respect to color. Of the color is the head direction. This neuron fires more or less in the direction of the reward to the right. This neuron has a trimodal response. It fires when it's going north, when it goes east, and goes west, but not when he's going south, like you saw before. And if you looked at the ensemble of neurons, they fire preferentially when the rat's facing that reward. This is the total, just like this plot, but across all the neurons. So they have abstracted out that the reward is roughly in this direction and roughly 50 centimeters away in this 2-meter maze. The third thing you want to know is the location of the reward. And indeed, when you take out these variables, the neural activity is concentrated around the reward zone and nowhere else, and that's a distribution across all cells. So they are firing exactly in the vicinity of the reward and nowhere else, once you take out these things. And the coolest part is that when you look at which neuron does what, majority of the neurons do all three things. Green are the neurons that encode distance, pink are the neurons that encode angle with respect to hidden zone, and blue are the neurons that represent the location of the reward. Neurons multiplex and do all the combinations. So this system is able to abstract out, and this is related to behavior, this is related to task demand. As they get better, these representations get better. As with experience, these representations start to predict rats' behavior. They start to tell if the rat's going to do better or not. I'm not going to show you these. Unless somebody is very curious, then I can show you later. I want to share one more little finding that we have, and then we stop. Any questions so far? So this kind of squared up the idea that there is some abstraction going on. It depends on vision and locomotion and other cues, so you better remove those things. And so far we went with very simple visual stimuli or very complex spatial tasks. Now we wanted to go towards completely the orthogonal direction, which is purely complex visual memory, which is what we are forming right now. Imagine that we take out sounds, then all you're forming is interesting impression about some complicated images. So what about that? Can we figure out how that is happening in this deep network? And this again is an experiment that people could have done. So we want to go back to the basics to say, since we solved this place cell, so hippocampus is required for episodic memory, but that patient HM, which is famous for brain damage, lots of movies are made about that, he could just sit there, he doesn't have to go anywhere, and still we have memory deficit. Can we do an experiment like that where there is no distance involved? There's no even moving bar of light could be invoking some kind of distance, so let's go away from distance. So we did this experiment. We leave these things aside, and we say, all right, let's take a purely black and white old movie. And it's intentionally touch of evil because what we are doing is quite heretic. So now it's just black and white movie, Hitchcock movie, a middle segment of the movie that clearly has no freaking idea what the plot is. Even you won't have an idea because it's right from the middle. And it's a continuous shot, so it's not like he's in one room and the camera is switched in these different rooms, so it's one continuous shot. So you should get nothing once again here, and just to get rid of space, we make it monocular. So big fat screen on one side, record on the contralateral side, and measure the activity in the entire deep network, all the stations, and show it 30 times. So let's see what the system does now, because now we are asking to do what kind of we are doing in this room. So we're going to show that activity. Here is the movie frame number. This is ancient movie, so 30 frames per second. So there are 30-second all, so 900 frames. You are going to see the spikes on the y-axis, 60 trials, and on the x-axis, here is the firing rate. And as expected, many neurons seem to care nothing at all. And they're kind of going around and boom, right there. Right at the end of the movie segment, this neuron pretty amazingly reliably fires. Here it doesn't fire, interesting, but otherwise pretty reliably it spikes at the end. So this fits with ideas when people showed movie clips to human beings and they changed the movie clip, neurons in epileptic patients' brain responded to the end of the movie when there is a cognitive boundary because the movie loops around. So even though this is one continuous shot, when this gets wraps around, then it's like a abrupt transition. So we thought maybe it's just that. And that would explain lots of studies in human, except this guy has no idea. Unlike in humans where they use famous movie clips of Bill Clinton's speech and this and that, he has no idea what's going on. So this is not like an overtrained, he has seen the movie only once. So that goes back to a question. He's seen it only once during training, and even then he has no idea. Then we look at another neuron, and now we start discovering other things. So it has very interesting structure, and the 2-second rule is gone. That previous neuron was active for roughly 2 seconds. This guy is now active for like 20 seconds with interesting structure in it. and also some evolution with experience, which is quite systematic rather than random. So, okay, these are all regular place cells. They're doing all this interesting stuff. Where is the abstraction then? We come to the final, the most interesting neuron. Quite shocking. That's what it does. Right? Pretty bizarre. Lots of bizarre things here. This part of the movie, the neuron likes. Spikes a lot in this part. This part, the neuron hates. The other neuron liked this part. It was spiking a lot in this part. Notice the amount of shutdown, right? Pretty much complete shutdown here. Then it once again likes. But then there is something even more surprising. There are some segments of the movie where this neuron likes that segment with extreme precision. This thing is just 2 movie frames wide, just 60 milliseconds. You're now 7 to 8 processing stages from the retina. If you got a response like this in primary visual cortex, you will say, yeah, maybe there was some interesting feature. But instead of that, 7 synapses away, this part, the neuron says, it's that thing. And then it shuts down and says, it's that thing. And then, on the other hand, pretty wide response. So we have a hint of what this thing is doing, but we are completely flummoxed. So at this point, we teamed up with Allen Institute, said, let's map the whole brain. Right? This is just a vision. So you put a bunch of electrodes, no reward, nothing is required. So you map pretty much the whole brain. So here's the LGN, where you have the center surround response, primary visual cortex, Gabor patches, higher order visual areas, first part in hippocampus called dentate gyrus, CA3, where Hopfield's work was done, CA1, where the place cells were measured, and subiculum that most people don't even talk about. The whole circuit, that's the farthest from the retina. So this is directly one step next to the retina. This is the farthest. And if you were aficionados of hippocampal or visual physiology, I would challenge you to tell me which is from which part of the brain. And most of us will fail. Yes, please. Okay. This is LGN. Hang on. Don't bet yet. Keep looking. Keep looking. Which one is LGN? I'm sorry to say what you said, LGN is not LGN, but that's okay. It's very similar structure called mega-scale coding. The same neuron in all parts of the brain, in some part, this is a neuron you already saw, so you know that's CA1 neuron. That's another CA1. This is CA3 neuron. This is dentate gyrus. This is subiculum. That is LGN. This is primary visual cortex. This is higher order visual area. You'll be really hard-pressed to tell me which is which. And yes, you might say LGN is highly active, but this is subiculum, and it has fairly similar structure. So can we now figure out this madness out of this, right? This is no cognition here. The machine is doing something. I can't explain with just dark and bright patches. I can't explain with Gabor patches. And hippocampus, I have no idea what the hell happened. There is no place here. So we started to think, okay, let's see if we can come up with some general features to explain this 500-fold selectivity difference. What is 500-fold? Just take any random neuron. Here the selectivity here is like maybe 100 millisecond, and here is like maybe 5 seconds. So it's like big differences. Here is a big amount, here is small. There are several neurons which is even more impressive, like this, 60 millisecond versus like 15 seconds. So big difference of selectivity, multi-scale or something.
But how are these panels ordered? So what is what?
Yeah, I was trying to challenge you, so I'm going to give you answer. These neurons are in LGN. Both of them are from LGN. These two are from primary visual cortex. These are from higher order visual areas, AM/PM. This is dentate gyrus. This is CA1, I think. I already screwed up. This is CA3, and this is subiculum. And those numbers, we have shown what they are in the paper, so you'll be forced to read the paper rather than rely on this. It's there, it's published last year. So then we thought, okay, is there any method to the madness? So we said, all right, let me just calculate the field duration. Different fields, some movie field is like 50 millisecond wide, some movie field is 5 second wide, and on a log scale, you see very large distance. So all these are hippocampal neurons. Notice on an average, the neurons in hippocampus like subiculum CA1, they have very wide fields on an average. But you still see some very 50 millisecond one, and you see giant ones. And this, on the other hand, is primary visual cortex. But then there are surprises. You would think, okay, right from periphery, you'll have tiny receptive field, and as you go further, the receptive field on an average will get bigger. But this is actually LGN. So primary visual cortex on an average has smaller receptive field than LGN. Weird. And LGN is more like higher order visual areas. And then some cool stuff happens in hippocampus, so things didn't go exactly as we expect anywhere. But here's a conservation law. So all right, instead of looking at the duration of individual movie field, I add up all the movie fields of a neuron. So total duration for which a neuron is active. This is the total duration, no longer log scale. All the brain regions on an average are active for about 10 seconds. So tremendous conservation law is appearing that every neuron in every brain region on an average prefers in a 30-second movie about 10-second duration. Notice the spread here is very small compared to this, where this is a log scale going several orders of magnitude. So there's some conservation law that's going on, which I don't know where it's coming from. And if you looked at how many fields there are in the neuron, if you allow me to use the word, if these are tokens, then what are the number of letters in the token? Very nice segregation. These hippocampal areas have very small number of words. They have only 1 or 2 or 3 movie fields per neuron. But each field is big, so they have the same conservation law. Whereas visual areas have tons of movie fields, sometimes as many as 15. And once again, this weird dependence, primary visual cortex has the largest number of these movie fields, and then LGN, and then V1, higher order visual area, and then there's a big jump. It's not the other way around. So some funny stuff is going on, which I can't explain based on this. We are working on it. Let me end. Am I pretty much out of time, or?
Yes.
Am I?
Any notes?
Sorry?
You had questions during your talk, so I guess.
Okay, I'll wind up. Let me just show you one final thing. So let me skip this part. The broad to narrow, you can look it up. There's an interesting conservation law there as well. So we did one thing. Say, what about that episode? I started the whole talk by saying there is some episodic memory. What the hell is episode? How do I define it? So all right, let's do a simple experiment. You take the same images, and you simply scramble them up. So each image is the same information as the real movie, but you put them in a random order. So now episode is gone, visual information is more or less the same, and we repeat that same random sequence many, many times, just like the movie is repeated many times. So here's the response of a neuron in primary visual cortex. X-axis is same, 900 frames, y-axis is firing rate. In red is the activity of the neuron to the continuous movie, and in blue is the scrambled movie rearranged to match the same real movie sequence. And what you see is that in primary visual cortex, the response to the continuous movie and the scrambled and rearranged movie is more or less the same, correlation 0.86. So primary visual cortex doesn't seem to care, and same thing is true for LGN and so on. They don't seem to care of the exact episode. Hippocampus, on the other hand, completely gone. Here is a CA1 neuron, very strongly tuned to the continuous movie, but once you get rid of it, no matter how much rearrangement you do, there is no selectivity left. So we have gone from like just Gabor patches and so on to something interesting. We feel this thing is doing something about continuity, so we computed so-called episodic movie index. We computed the selectivity of a neuron in z-scored unit to the continuous movie minus the scrambled movie divided by the sum to say this is how much the neuron cares for whether the movie has a continuity or not. All the visual areas, their selectivity index is kind of close to 0, which means they don't care whether it's continuous or not. Once again, funny thing, primary visual cortex is right here. That cares the least for whether the images are going continuously or scrambled. LGN cares more than primary visual cortex substantially whether the images are continuous or scrambled, higher order visual area, and then hippocampus is pretty much 100%. So this thing kind of tells us a bunch of things. And I'm just going to end with here. I didn't explain all this, so don't worry about all those things. So immobile hippocampus reliably encodes purely visual episodes without any task demands or rewards. And if you include rewards, then it starts to do this weird multiplexing and so on. So I'm happy to take any questions. Let me just stop here.
Thank you.
Thank you.